I will be changing topic a little bit from what we've been hearing before. And for that, I would like to ask you if you have ever wondered how many muscles do you actually use when you walk or you swim or you run, for example. If not, if you do a quickly Google search, you will see that this is around 100, some hundreds of muscles that represent actually around the 30 to 40% of our total number of muscles in our body. Which indicates that actually motion, it's very important in nature, and not only for us humans, but also for other living organisms like animals or plants. And it allows them to communicate with each other, look for food, mate, protect from predators, for example, or carry out tasks that they couldn't do anyhow alone. And it's not only in the macro scale, but also in the nanoscale or at the subcellular level, where motion allows for reproduction, cell division, or even transporting cargo inside a cell. And this motion at the nanoscale has also inspired science fiction, movies, or Netflix shows, or like the example that I bring here today, that is from the Marvel universe, where this superhero, the Ant-Man, is able to shrink to the millimeter scale. But even when he does that, he preserves the same kind of motion that he would have at the macro scale. So he can actually move as fast as if he could be moving in our regular size. And this is because usually this kind of shows omit or don't take into account the problems of motion at the nanoscale. So motion can be defined by the Reynolds number, that is basically the ratio between the inertial forces that play a role in the macroscale and the viscous forces that govern at the nanoscale. And if we try to reproduce, for example, the motion of a fish that has a Reynolds number of higher than some hundreds of thousands, we can do this in the lab using this kind of device, where we put the device into a bucket of water and then it flips, as it would be the flipping of the tailfish, of the fish tail, sorry, in the ocean. And if we now want to study how this fish could move at the nanoscale, we cannot shrink the fish, but what we can do is to change the viscosity of the media from water to, for example, honey or some syrup that would increase the viscosity around the fish. And then, as you can see, using exactly the same device, the motion is not permitted anymore. And if we take a look now at the motion at the nanoscale or the macroscale with some organisms where the Reynolds number is below 0.1, we see that they have different mechanisms of motion that are actually non-reciprocal in time. That means that if we change now our device and we use, instead of a flapping tail, we use a screw-like shape, even in the same conditions, we will be able to actually see motion of this device. And this has been done during the last years by scientists designing devices that can move in the nanoscale. And this is what we actually called nano or micromotors, or that also called streamers. And these motors are self-propelled particles that can rate Brownian motion by converting any kind of input energy into kinetic energy. They are basically classified into two groups: those that use external stimuli for locomotion, like magnetic fields, light, or ultrasounds, and those that actually use that are powered by conversion by biochemical or chemical reactions, converting substrate that they can find in the nearest environment into products. And by doing so, they can also sustain locomotion. So in the laboratory for cell mimicry, where I work now as a postdoc, we've been doing this during the last years. These are only some examples of what I've been doing since I joined the laboratory, with different kind of motors that also move in very complex environments. And today I will only focus on two of them that would employ magnetic nanoparticles actually to accomplish locomotion. The one thing that we have to take into account is that since these motors appeared 20 years ago, they went from very simple aqueous environment into more complex media like cell media or even inside cells, also gels, and eventually we can see examples nowadays of motors moving inside organs like a stomach or intestine. And together with the advancements or with increasing the complexity of the media, we could see that also the complexity of the motors was increasing over the years. But still, one thing is that all these motors have shown locomotion in liquid-phase environments, like water and some kind of fluids. And the question here could be, can we actually make motors that move in environments that are not liquid-phase? And for that, I bring an example where we use a magnetic motor, but we use different 3 types of them, but I will just focus for the interest of time in one of them. And we move from the motion in water to the motion at the interface of cellulose, water, air. So we basically put these motors into paper chips and see if they are able to move. So the motor we are talking about, they all consist in this, they all consist in polystyrene bits that are decorated with magnetic nanoparticles, iron oxide nanoparticles by a layer-by-layer technique. And then eventually we will add an extra layer of a fluorescent labeled polymer in order to visualize these motors. And this is an example where we use a 500 nanometer core or motor, but we also do it with different sizes. And in all of the cases, what we're interested to see is that these motors are super paramagnetic and they respond very quickly to the magnetic field. And this is a very simple but also very efficient model to study locomotion in different or more complex environments. So we can actually focus on seeing the locomotion, exploring the locomotion, and not in the motor design as such itself. So we take these motors, we put it, sorry, it's because I have this thingy here, so you probably see the panel above the slide. So you can see now how these motors look like on the surface of a paper chip when they are false colored in pink. They are not able to penetrate longer into the paper layer, meaning that they are actually moving on the surface. And then what we do is we apply a magnetic field of different field intensities or magnetic field intensities, and we record the locomotion of these motors. And as I hope you can see here, where no magnetic field is applied, the motors cannot move. These are these small dots in black. And then when we increase the intensity of the magnetic field up to 3 Newton, then these motors move in the direction of the magnetic field. And the trajectories are longer and faster when the magnetic field intensity increases. So from here, we can actually track every independent motor and then plot or allow for calculation of some specific features, like, for example, the velocity. And we do that, and you can see how the velocity, of course, increases when we increase the intensity of the magnetic field. And this has been done for a motor of a size of 4 microns. But if we increase the size of the motor, of course, the response to the magnetic field will vary. And it will vary according to the magnetophoretic mobility coefficient, where it scales with the radius of the particle, also with the magnetic susceptibility, and then it decreases with the viscosity of the media. So this is an example where we increase the motor size from 4 to 40 microns. And obviously, when we apply the magnetic field, we have motion, like a higher velocity compared to no magnetic field. And then, of course, when we increase the size of the particle, then the velocity decreases. But an interesting point here is that when we use low concentration of particles for the same conditions and high concentration of particles, there is not much of a difference up to 10 microns in size. But for the larger motors, they actually move faster when high amounts of particles are being considered. And this is because of the dipole interactions between the particles that actually overcome the viscous or the resistance of the motors to move across or along on the surface of the paper chip. And this was then used actually in an application for my colleague, Paula de Dios. She's a PhD student in the group. And she used these paper chips or these motors to allow for the motion of keratinocytes, that are skin cells that help to heal the wound when we have any kind of wound in our skin. So she put these motors together with the keratinocytes, and she allowed the keratinocytes to just move autonomously and interact with the motors first. And after this was accomplished, then she applied the magnetic field in a paper chip resembling a wound model where there was a break, and we can see the motors are here standing in red and the cells in green. We can see how the motors start to fill the gaps in between, because it assists, they assist for the migration of the cells more efficiently compared to when no motors are considered. Sorry, as you can see in the above panels. So in this case, we have, can I just remove this? Yeah. In this case, we have sizes of motors that are similar to the cell size, and then they don't penetrate inside the cells. But some other times we might want the motors actually to penetrate inside the cells. As this is an example from my PhD that I did with Professor Veronica Salgueirinho and together with Monica Lopez Fanarraga from Spain, when we see that the particles actually penetrate inside the cells, and if they are endocytosed, if they are eaten, let's say, by the cells, they can move freely in the vesicles where they are internalized. But if we apply a magnetic field to pull the particles inside, they get trapped in the cytoskeleton of the cell, and they are not able to move any longer. So a question arose here that was how these motors actually are able to cross these cell membranes. And for that, we came across a model where we resemble a cell membrane by using a giant lipid vesicle, where we change the mechanical properties of the lipid vesicle, and we put it in contact with the magnetic motors to see how they penetrate or go through these lipid vesicles. So these lipid vesicles, you can see them here, are made by a double emulsion transfer method, and now the lipids are labeled in green and red, and we change the mechanical properties of these vesicles by changing the lipids, that the building blocks that form part of the vesicles are made of. So if we use this DOPC, the gray lipid, we will end up with flexible zwitterionic membranes that we can change the surface charge of the membrane by adding the blue lipid, DOPS, that will enable to make flexible but negatively charged membranes. And then we can change the flexibility by changing DOPC by DPPC in green, that is a fully saturated lipid, and then it will make stiff, very rigid membranes that are also zwitterionic. And then we can account for the mechanical properties of these membranes by using the Loudon probe. This is a small organic molecule that can intercalate into the leaflets of the membrane vesicle, and then it changes the emission. It's a fluorescent molecule that changes the emission according to the fluidity of the membrane. So very low values or negative values of GP, that is the general polarization of this molecule, will tell us about flexible membranes, and then positive values will tell us about more rigid membranes. So now we have our membranes and we have our motors. We put them together, and I hope this video works now. Okay, it doesn't. It's because we are having some trouble here migrating files during the last days in the university. So probably the video is not longer and in the same location. But what we would see is that the motors, some of the motors are able are able to penetrate the vesicle and get trapped inside. Some others will get stuck on the membrane of the vesicle and cannot go further, and some others will even not interact with the membranes. And this will give us an example or a representation with different kinds of motors with different velocities, because the velocity will be different if they are moving outside of the vesicle or inside of the vesicle, because of the difference viscosity. Outside is just water, inside is a very viscous environment. And we cannot just account for the average of these motors to, for example, calculate the velocity or the energy that they need to cross the membranes. And we need to come up with a strategy to actually classify these motors according to what they are doing when they interact with the membranes. So with the help of Andrzej Hovorka from the University of Southampton, we developed a mathematical analysis model that what it does, it is able to discriminate between these groups of motors. So this is an example of a trajectory map that I would get after the experiment. The circle would represent a vesicle, and then all the dots are the motors moving in the direction of the magnetic field. So for example, motor 1 would move only inside of the vesicle, while motor 2 will be moving outside of the vesicle. Then motor 3 is able to cross, and then finally motor 4 is stuck on the surface of on the membrane of the vesicle. So one thing that we can do to actually discriminate or classify these motors into the 4 groups is to analyze the velocity plots. So if we have a continuous velocity with a steep slope, this tells us about motors that move continuously and with fast speeds. And those would be motor number 2 moving outside of the vesicles. Whilst if we have a continuous velocity, linear velocity, but with a smaller slope, this will tell us about motors moving continuously but slow, as it would happen for motor 1 inside of the vesicles. And then these able to cross the vesicles would be those having first high speed and then slower speed. And then for this would be number 3. And then for motors number 4, we would have initial velocity that would go down to 0 when it touches the membrane. And then we can actually plot this as a pie chart, where we have here in different colors, like the motors moving inside in orange, outside in purple, those crossing in yellow and getting stuck in blue. And we even account for motors doing something else that we cannot control, and they are classified in the gray group. So for the next slides, we will only focus on those motors, like the yellow group, that those are the motors able to cross, because they will actually give us some information about the mechanics and the mechanism for crossing the membrane. So first, we put our motors together with the zwitterionic flexible membranes, and we record experiment for 3 minutes. And this is endpoint images from the beginning and at the end of the experiment. And first thing that you can see is that the motors cross and get accumulated inside of the vesicles. It's very important to say that they don't break the vesicles. So there must be some stability or some flexibility of these vesicles that actually they don't burst when such a high number of motors are crossing through them. And also a second thing that I would like to mention or highlight is that the model that we developed is able to calculate or to classify more than 300 motors in a sample. Whereas if you take a look at the current literature, they usually account for some tens of motors. So now if we do our plotting, we see that around 34% of the motors are able to cross the membranes. And then we can take this piece of the pie chart and analyze how these motors cross the membranes. And if we do that by counting the normalized pixel area, so counting the number of pixels that get accumulated inside of the vesicles, we will get a plot like this, where we have, after some time, a plateau, meaning that the vesicle is completely, or no more motors will cross the vesicle. And this follows a logistic function that has this shape. And this function allows us to actually calculate 2 important parameters here that are tau and p. So tau is the permeability, the permeation time, so the half time that the motors need to cross the vesicles, whilst p is the permeability of the vesicles towards the motors crossing. And those 2 parameters are specific depending on the type of motor and on the type of vesicle. And we can plot them and then compare to our next example. So our next example would be using a negatively charged vesicle, where you can see a similar behavior, but then you can see how the number of motors that are able to cross increase up to 41%. And this is because our membranes are now negatively charged and our motors are positively charged. So there is an interaction between the membranes and the motors that actually make easier the motors to cross, like to interact with the membranes and then to cross inside of the membranes. but they follow a similar profile while filling up the membranes with a specific permeation time and permeability. And our next example would be use instead of a flexible membrane, use a rigid membrane. In this case, we also change the magnetic field intensity. We go from 2 Newton, this is what we've been doing before as well, to an 8-time lower magnetic field to see if it makes any difference when crossing these rigid membranes. So first for the 2 Newton, so the same examples as before, we have an increase of up to almost a 50% of the motors that are able to cross the membranes. And this is because now if you think about these membranes and the other membranes, so these membranes are more rigid compared to the previous ones. And this is like if we try to shoot a football ball to a camping tent or to a window glass. So if we do it against a camping tent, this is more flexible and it allows to dissipate more energy. And then it's more difficult to actually cross or to break through and cross to the inside. Whilst if we have a window that is more rigid, it cannot dissipate energy that much. And that makes easier that every time the motor impacts in the membrane, it will cross through. And we got similar trends for the filling rate. And now when we change to the weaker magnetic field, we have even a larger number of motors able to cross. And this is just because these motors are now slower because the magnetic field is weaker. So they move slower and they have now more time to interact and the interface with the membrane and actually be pulled through, as you can see from the data here. So just to wrap up, I would like to show you how these membranes look like after interaction with the motors. I just took one example of the zwitterionic flexible membranes, and the membranes are labeled in red and the motors in green. And you can see we have again the accumulation. And if we do a line scan through the vesicle, you can see how here the green and red signals overlap, meaning that probably our motors are actually carrying part of the membrane or the lipids with them while crossing. So this could resemble a synthetic phagocytosis model where we have our motor that encounters the membrane. It's still pulled because of the magnetic field, so it starts to wrap, or the membrane starts to engulf the motor and then wrap around it until it pinches off, and finally it gets internalized with a layer of lipids on their surface. So this is probably what is happening here and why we see this overlapping signal. So from this, the next obvious step would be to calculate the energies that these motors need to cross the membranes. And then afterwards, this would be also used not only for magnetic motors, but for other kind of motors and other kind of membranes. That would be a lot of applicability when using these motors to interact, for example, with cells, with biological membranes, and to see what's the minimum energy requirement that these motors need to actually cross through the biological membranes or biological barriers. So with that, I would like to thank the group, the Laboratory for Cell Mimicry. Special thanks to Professor Brigitte Stettler for giving me the opportunity to work with her, and Paula de Dios for sharing the slide with the keratinocytes. Also to Professor Andrei Hovorka for the analysis, or the help with the analysis of the motion and the founding bodies. Of course, I would like to thank the organizers for giving me the opportunity presenting my work in this conference, and all of you for listening. And if you have any kind of questions or comments, they are very welcome. Thank you so much.
Thank you for your fantastic talk, Miguel. I'm sure there are questions from the audience. Please raise your hand or write in the chat.
And it's a very out of topic almost. It's more like using the magnetic particles for application.
I see. Well, while people are thinking, I will ask a quick one.
Yes.
So since you considered membrane with different rigidity, which type is actually closer to the ones which would be in real tumor cells to be able to penetrate, or it really depends on the type of the tumor?
Yeah, no, that's a very good question, because here we are looking at a model for a single cell. when we have a tumor, what it happens if these cells also originate an extracellular matrix around that becomes thicker, and it's usually made of collagen in most of the cases. And it's a very interesting question because that's part of the research that I'm carrying out right now with another student. But instead of magnetic motors, he would use collagenase. So it's a motor that has the enzyme on the surface that is able to eat this collagen to see if we can penetrate, and then apply a similar model to see how much energy we would need to actually do that. I'm not aware about if these magnetic motors would be able to cross such a thick barrier, because they are not super. Once they encounter the barrier, they also lost a lot of energy, right, in the impact. So far, I cannot answer that question, but I hope in the near future we can have some solution for that.
Thank you. That sounds great.
Yeah, thank you.
I cannot see any questions, but we will now move to the gather town. And Miguel, maybe if you could stop by there for some minutes so that participants can catch up and thus continue.